27, 1822, in Dole, in the Jura region of France. I lived just after the period of Industrial Revolution. The curse of industrialization in France was so idiosyncratic that for a long time people wondered whether an industrial revolution had ever taken place in the country. Even though the change wasn't that radical in France, scientists like me had much more machines or artifacts to develop their experiments. There was a large variety of highly developed traits in 18th century France. Thanks to this, diseases were more common everywhere, and this is what motivated me to investigate further into this experiment. I was a French scientist, and my first vaccine discovery was in 1879 with a disease called chicken cholera. After accidentally exposing chickens to the attenuated form of a culture, I demonstrated that they became resistant to the actual virus. I went on to extend the germ theory to develop causes and vaccinations for diseases such as anthrax, cholera, TB, smallpox, and rabies. In rabies, I began the course of 13 injections, one each day, of vaccine made of rabbit nervous system tissue. Each successive injection contained a less attenuated virus. Mr. never developed rabies, and the incident was regarded as a success. At that time, there were a lot of poor people due to the wars that Napoleon was fighting. People didn't have money to pay for their treatment, and also rich people that needed to be cured. So I had the money and time to research about the diseases, and my first important discovery in the study of vaccination came in 1879 and concerned a disease called chicken cholera. However, they had to wait 80 years until I first isolated and grew it in pure culture, because the disease was first recorded in 18th. The same year, I continued my research in the vaccine, when France went through an anthrax epidemic. An outbreak of anthrax threatened France's livestock, especially cattle and sheep. I produced vaccines from weakened anthrax bacilli that could indeed protect sheep and other animals.
The effects were very positive because it was a new invention that could cure people, more lives would be saved, and the rate of death could be decreased. It was very useful that later the scientists around the world began to dedicate into that subject and to make more experiments to discover how vaccines in certain type of diseases. The idea of having a vaccine to treat a disease nowadays is still used in medicine because it was a very good idea that it was made into full research, and a lot of doctors and scientists have discovered and improved the vaccines. New techniques for vaccines are made each day, like DNA vaccines and the recombined vaccines, DNA. Vaccines consist of DNA coding for a particular antigen, which is directly injected into the muscle. Like recombinant vaccines use attenuated viruses or bacteria strains as vectors. A virus or bacterium from one disease essentially acts as a delivery device for an immunogenic protein from another infectious agent. In some cases, this approach is used to enhance the immune